All right, well, here's our lecture for class tomorrow. Sorry I didn't get it in last night, but here we go. So it's the remainder of Chapter 10, and as a reminder, we're going to spend a little bit of time in class talking about some of the statistical tools that we use to model the relationship between the genome and complex diseases and the expression of complex diseases. In this recording, we're really just talking about the remaining biotechnologies that are used as we study these. All right, first key term, exome sequencing. And so we've talked about some of the issues with the sequencing the entire genome. One is that we have gaps in the genome that haven't been fully sequenced. We have an enormous amount of variation, but we have mostly just the same information from one individual to the next. Most of the genome does not vary from one individual to the next. And so it's so much information, and being that's a lot of that information doesn't vary from one individual to the next, in essence, for some of these population level studies of complex diseases is useless. And so we don't really use full genome sequencing to do these population studies. And what we might do is what's called exome sequencing. And so in exome sequencing, this is where you sequence just the coding genes. Keeping in mind, most of our mutations that are associated with monogenic genetic illnesses are in coding genes, which should make sense. However, most of them that are associated with complex genes, complex diseases, are outside of coding genes. However, we do have those rarer mutations that have large effects or a large ability to predict the occurrence of one of these complex diseases, and those mutations are often in coding genes. So this is valuable even for the complex diseases. DNase-1. So DNase-1 is an enzyme that basically targets specific regions of the chromosome that have certain protein signatures. And so DNase-1 is used to basically just sequence what we would call the DNase-1 hypersensitivity sites, which these are going to be sites that are actively transcribed. This was the technique used during the ENCODE project to basically figure out what the other 98% of the genome is doing if it's not representing coding genes. We found that 70 to 75% of it is found in these DNase-1 hypersensitivity sites, which is an indication that it is actively transcribed. CHIP sequencing. CHIP stands for chromatin immunoprecipitation. And then the sequencing is followed by sequencing. And so here we sequence just regions of the genome where we find a specific protein. So what we'll do is we'll design an antibody against some protein. Maybe it's a transcription factor, maybe it's a enhancer, maybe it's an enzyme that works to rearrange DNA between heterochromatin and euchromatin. Regardless, we design an antibody against a protein that binds to the DNA molecule at some location. And then we will, where we find those antibodies binding, we will sequence those sites. And so it's a tool that we can use to figure out which genes are going to be transcribed or upregulated as a result of the same transcription factor. So if we know that, in some cell signal, it leads the cell to produce more of this particular transcription factor. And we want to know, okay, well, what are all the genes that are going to be transcribed then as a result of the upregulation of that transcription factor? This is what we do. We design an antibody for that transcription factor. We find all the places in the genome where that antibody is binding, which is an indication that that protein is binding the genome at that site, and then we sequence that site and figure out all of the genes that are going to be transcribed again as a result of the upregulation of that transcription factor. RNA sequencing. So this is used to do gene expression studies where we're not sequencing the genome, we are instead sequencing whatever RNAs are present, whatever RNAs are actively being transcribed. And we can target this to very specific RNAs so that we can determine whether or not a particular gene is being transcribed or being expressed, and how much of it, relative amounts. And so RNA sequencing is a very valuable tool for doing this, although it is a little bit more time consuming and a little bit more labor intensive than some other technologies that are becoming the standard, if you will. One of those technologies is a microarray. So in a microarray, we basically take a solid surface, usually a slide or something that looks like a slide, and we put little tiny beads of single-stranded DNA on it. And the single-stranded DNA is basically complementary to certain target RNAs or certain target even cDNA. If we take, all of the RNA we find in a cell and then we induce that RNA to develop cDNA from it through reverse transcription. But anyways, we take this solid surface that has all these little beads of DNA that are complementary for certain target nucleic acid sequences. And then what we'll do is we'll take all of our we'll isolate all of the nucleic acids from a cell and we'll place them on this chip. And if it has some of that target nucleic acid, it's expressed some of those genes, and it's making that RNA, then that RNA will bind to the little beads of DNA that we've put on that solid surface that are complementary to our target nucleic acid. And so this is a very effective way in which we can determine whether a specific cell is expressing a specific gene. And it's very quick. When it first came out, it was very expensive, but now the cost is lower, and it's just a lot less labor intensive and complicated than doing RNA sequencing. And so again, it's becoming one of the main ways in which we do gene expression work. All right, so the last part of Chapter 10 is about animal models, using animal models to study these genetic diseases. And so rather than just doing these hypothesis-free analyses or these hypothesis-free studies, what we'll do is we'll actually do a hypothesis-driven study. But in order to do that, to avoid ethical concerns associated with changing the genetics of human beings, we do animal models. And so one of those model types is called the knockout models. And here it sounds exactly like what it actually is. We knock out a specific gene, and then we look at the phenotypic effects of that knockout gene. So these are called knockout models. Next one, transgenic models. These are in some ways the opposite of knockout models. Here we basically create a transgenic animal that is typically going to lead to the overexpression of a particular gene, or the expression of the gene for the first time if that animal doesn't actually express that gene. And so here, again, we use this in animal models to figure out what does this gene actually do when it's expressed, and we get it to be expressed in a really high level so we can see the phenotypic implications of whatever that gene is. And so, I don't know why that one's still on there. That one's left over from the previous slide. You can ignore that last term. And so these are the key terms that we're going to use to build the foundation for our class on Wednesday. Hope you enjoy.